Okay, here we are back at the signal peptide, and we are talking with endoplasmic reticulum. We're trying out the animations here. So let's get started. Boom, 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 boom. Okay, so let's look at topic number 19 today. So topic 19 is covering recombinant DNA technology and forensics. There's a ton that could be covered in this topic, and every semester I usually make it a little bit different, looking at different examples of things that have been done. Mostly in this topic I want to go over some important technologies and a few brief examples. I'll try to keep it short. I'm truncating most of my topics at the end here just to sort of cover the basics on what I think you need to know for moving on into second year. So first, let's talk about the DNA toolbox. What do I mean by this? I want to talk about some tools and technologies. And the good news is we've actually talked about most of these, at least in a small amount of detail. So here are some of the tools of the DNA toolbox. You can see PCR, restriction enzymes, gel electrophoresis, plasmids, vectors, CRISPR, and Cas9. So let's talk about those. So let's talk about PCR. I talked about PCR very briefly. If you go back to our unit on DNA replications, I talked about PCR. PCR stands for polymerase chain reaction. So you're probably thinking polymerase. Which polymerase are we talking about? We are talking about the DNA polymerase. That is the one we're talking about. And what is a chain reaction? A chain reaction means you do something again and again and again. So this is basically DNA replication in a tube. So how does this work? Well, what are we putting in that tube? Well, you need your DNA sample. So if this is forensics, that DNA sample could be a single hair from a crime scene. Or if you're trying to clone a Tyrannosaurus rex, it could be some DNA you found in a fossil. We need some primers. So remember, when we were doing DNA replication, we had an enzyme primase that made some RNA primers. In this case, these primers we can get made by a chemical company. So we can actually, you can go online and you can order them, and a chemist will make some primers for you. And these ones actually won't be RNA, they'll be made out of DNA. What else do we need? We need nucleotides. So which nucleotides are these? These are the D, N, T, Ps. That's dinucleotide triphosphates. So remember what those are. We've got the A, the C, the G, and the T, and those are the substrates for the enzyme. So don't forget that polymerase. There's our enzyme, our DNA polymerase. There it is. Notice this one here is heat stable. So we got it from some sort of thermophilic organism, and the reason why we need a heat stable DNA polymerase is because we're going to heat up this sample quite a bit. So we'll talk about that in a moment. Quick test question for you. What does DNA polymerase do? Does it copy the sense strand? Does it copy the template strand? Does it add nucleotides complementary to the sense strand? Does it add nucleotides complementary to the template strand? Does it make primers? Let's start here. Nope. Primase makes primers, or a chemist in the case of PCR. Does it copy the sense strand? No. Does not copy the sense strand. Does it copy the template strand? No, it does not copy the template strand. No, it does not add nucleotides complementary to the sense strand. It adds nucleotides complementary to the template strand. So remember, the enzyme actually looks at the template strand, and the DNA it makes is a copy of the sense strand, but that's not what the enzyme does. The enzyme actually looks at the template strand. So just go back and take a look at DNA replication. Please do that. That will be a huge part of your final. You need to understand DNA replication. If I have time, I will go back and redo that lecture in an online format so everyone has all the information for that. So once you have these things in a tube, what do you do? So here we go. We throw all those things in the tube. We have our target DNA, so that could be, like I said, a hair from a crime scene. And what are we going to do? We're going to heat that up. We heat it up to about, I think it's about 94 degrees Celsius. and that will break all the hydrogen bonds. We don't need a helicase or anything like that if we use heat instead. The next thing is we throw those primers in there. The primers will recognize the gene of interest, so we could be looking for all sorts of genes. We could be looking for the gene for hemoglobin. We could be looking for the gene for insulin. It doesn't really matter. It depends on what you decide as the scientist. After that, we let the polymerase do its job. It adds the new nucleotides, and now we have 2 copies of DNA. So what do we do next? We do this again. Now we have 4 copies after 2 rounds. After 3 cycles, we end up with 8 copies. And you can imagine if we do this again and again, we get 16, 32, 64, 128, 512. And if you do about 20 cycles, you end up with about 1 million copies of DNA. Most people do 25 to 30 cycles, and you end up with much, much more. So 1 copy of DNA, 1 hair from a crime scene, and now you have a whole bunch of DNA for analysis, and you can determine whether you have found the perpetrator or not. So what is PCR used for? So of course it is used hugely in genetics research. Any gene that I'm interested in, I can make copies of it and study it and research it. That's a big part of what I did in my PhD work. You can talk to me about that anytime you like. I mentioned forensics and paternity testing. I will be going over that in a little bit of detail in a few minutes. A new area where PCR is being used actually is archaeology and paleobiology. So we've extracted DNA from mammoths and mummies and all sorts of really interesting things. And of course, there's lots of medical uses for this detection of pathogens. So this new SARS virus, the COVID-19, is actually a PCR test. We're actually looking for viral RNA in people's saliva from the swabs that they're doing. Because the virus itself, we can't grow it yet in a lab very easily, or that's not actually a routine thing to do. And so they're actually looking for the viral RNA by using PCR test. So another tool is cutting and pasting. So cutting and pasting is using these particular enzymes called endonucleases. So remember we talked about endonucleases as being enzymes that are part of the replication and DNA repair machinery. And these restriction endonucleases, these are actually enzymes, you can see it says here, they cleave DNA at specific sequences. So take a look. Here's a sequence here. This is AGCT. So notice AGCT, 5' to 3'. If I look at the other strand, if I read 5' to 3', it's AGCT. So this is called a palindrome, kind of like the word Bob or race car or something like that. And this is where the enzyme recognizes. So these enzymes, here it goes, it's going to cut it. You can see I cut the DNA right here into 2 pieces. So here's another one. This is EcoR1. This one comes from E. coli. That's what the eco is for. And you can see it recognizes GAATTC. And there it cuts. And so these particular enzymes are found in a variety of organisms. And there are companies that have them. And they've extracted them, purified them, and you can buy them in little test tubes. And these are like precision laser scissors, where they're cutting very specific pieces of DNA. They're used all the time in genetic research and biotechnology. I'll show you an example on the next slide. So here's how it might be used. So notice there's the GAATTC. We can cut that DNA. There it's cut. And we can grab another piece of DNA. So here's another piece of DNA that is cut by the same restriction enzyme. And now we can put them together. And there we go. And now what we have here are 2 pieces of DNA that have been put together. There's still little gaps here, right here and a little here. We can use our DNA ligase from DNA replication to put the whole thing together and make some recombinant DNA. So what are some of the applications of this? Many, many applications. The one we have talked about a few times is, of course, where we took the gene for human insulin and put it in E. coli to produce insulin for diabetics. Another very important tool is agarose gel electrophoresis. So you talked about this in your recent lab report, so hopefully you know a little bit about how it works. So the gel part here, this is the gel right down here. And we'll grab another color. Here's the gel. We'll color it in. The gel is made of agarose. Agarose is basically a carbohydrate extracted from seaweed. It's jellified, so it's heated up with water and makes a matrix. The DNA is loaded in the sample right here. The DNA is negatively charged because of all those phosphate groups. It runs towards the positive electrode and separates out by size. So here's a DNA gel electrophoresis, and you can see all sorts of bands of DNA that is separated out by size. Notice down here we have some smaller fragments. Up here we have some larger fragments, and this is an important part of a lot of analysis of DNA products. We can also have RNA gels. So you can see in this case here, somebody's looking at different RNA levels in different tissues, and useful for, of course, people who are doing RNA research. We can also have protein gels. So this is a different technology, but this actually is one of my research gels. I'm looking at a protein called penicillin binding protein 2, and you can see that I've purified it. Not perfectly pure, but you can see I have it there in large quantities, and I'm analyzing it on this particular gel electrophoresis apparatus. So one piece of technology that has received a lot of press is this one here. It's called CRISPR-Cas9. I do not have the time this semester to go over all the details on how this works. If you want a bit of more information, I encourage you to check out this video. I will put it in the course playlist, and it goes over in a little bit of detail on how it works. The huge thing about CRISPR-Cas9 is this can do DNA editing, and it can do it inside the cell. So inside cells. There we go. So this is, of course, we can change DNA inside cells, and so the potential on this is huge. I will talk about a few CRISPR-Cas9 projects at the end of this lecture. Here's a slide that takes a look at how it works. You can take a look at this in the textbook. This is figure 20.14. Like I said, I'm not going to go over this in any detail. So let's talk about some applications and a little bit about implications on this. This is something that's very hard to cover in an online lecture. It's much easier to discuss this kind of thing in class, but let's take a look at some examples that I've come up with. So first part is forensics and paternity testing. This has really changed, obviously, crime investigations in many cases, and it's a very, very important technology. Let's take a look. So forensics and paternity testing, there are a few methods, but the most common modern method is something that is making use of VNTRs. So VNTRs, you can see up there, stand for variable number tandem repeats. So what are these things? Well, it turns out that everybody has these parts of their DNA where you have these little sequences that are repeated. So that's where the repeats come in. And some people have repeats, maybe 10 repeats, some people have 100. And it turns out there's a huge amount of variation in the number of repeats that a person has in the population. So because there's a bunch of differences amongst individuals, this is something we can take a look at and can be used for DNA fingerprinting, meaning that we can look at differences between different individuals. So take a look at this example here. We have individual 1 over here on the left. And individual 1, we're looking at allele A5. So this particular person has 2 alleles. Remember, we get 1/2 of our DNA from mom and 1/2 of our DNA from dad. One of their parents gave them 5 repeats, so 1, 2, 3, 4, and 5. The other parent gave them 2 repeats, 1 and 2. Individual 2 has a different pattern. They have 3 repeats on one chromosome and 4 repeats on the other chromosome. So if you take a look at a gel of these kind of things, different individuals have different patterns. So take a look, this is a gel of 6 different individuals, and they all have different patterns on that gel. Now, there is a chance that 2 individuals may have a band that's the same or similar. If you take a look, for example, I'm looking at this individual here has a band right there, and individual number 6 has a band there that are very, very similar, and this kind of thing happens all the time. So we're not just looking at 2 alleles. In most cases, for crime scenes, we're actually looking at 13 alleles, and so the probability of having 2 individuals that are identical is very, very low, unless, of course, they are identical twins, which will have the same DNA. So let's take a look at some actual examples of some paternity testing. So paternity testing is where we're looking to see if the father is indeed the actual father or whether he is not the real father or not. So this is a very simple example, but usually what we do is we have the mother's DNA. Usually we do know who the mother is, and the child's DNA. So take a look. The mother and the child should share 50% of the bands. So if you take a look, the mother here shares this band with the child. This band it does not share with the child because she's only giving half of her DNA to the child. The father should also share 50% of his DNA with the child, and it looks like that is indeed the case. If we look at the paternity being excluded, again we use the mother as the control. Notice the mother is still sharing 50% of the DNA. We want to make sure that the technology is working. And the second band does not match the father's. So in this case, this guy is not the dad. So here's a more complex example. Notice these are still cartoons. It's really hard for me to find some actual really good gels pictures. I'll show you a good gel picture in a minute. But the same thing here, this is the same idea, except for now we're checking multiple alleles. So on the left here, you can see we're checking, we have 50% of the alleles match with the mom. And on the right here, 50% of the alleles match with the dad. So in this case here, the answer is yes, this is the dad. Here's an example where the paternity is a no. Again, on the left, we're checking that the mom does match. And on the right, we're looking at the other bands, and no, they do not match the dad. So in this case here, the answer is no, and there is no paternity. So here's an example of a real gel, a real paternity test, and I'll give you something like this on the final exam, and there'll be a multiple choice question about it. So you'll have to answer this kind of question. Or the question might be, which of these children do not belong? Or are all these children belong to this father? Or who is the correct father? Or something like that. So take a look at this here. Over here on the left, we have the mom. Okay. And then the second person here, Luke, is the child. And so we're looking at this and we're wondering who the father is. So the mother, it looks like half the bands do match with the mom. That's good. Gets a little crowded down here at the bottom, but we do have consistency with the mother. We can look at Darth Vader, and right here it looks like yes, we got about half of those bands are matching with Darth Vader. So yes, indeed, Darth Vader is the father. We can take a look at Obi-Wan Kenobi, and it doesn't look like we're really having very much success with matching. Maybe right here, but again, like I said, sometimes there are some flukes just by random chance, but you've got to be matching 50% of the bands. So take a look on the final exam for one of these type of questions. They should be very easy to interpret, and now you know how to answer these kind of questions. So of course this type of technology is used for forensics testing and is a very, very important part of investigations nowadays. Not all the time. It's expensive, so they're not using it all the time, but there are some people that have become released from prison due to this. Here's an example from the textbook. This guy's name is Earl Washington. He was in prison for 17 years. And notice they had a chance to look at the DNA evidence. So in this case here, they're looking at semen on the victim, and they're looking at 3 markers. So here are the sizes, 17, 19, 13, 16, 12, and 12. They looked at Earl Washington, no match, no match, no match. Then they went and took a look at a second suspect, and this guy here, it turned out he matched perfectly, and so they released Earl and arrested Kenneth. So like I said, this is done by all sorts of criminal investigations nowadays. A big part of it was developed in the United States by the FBI. It's called CODIS. You may hear about that in crime shows. The Canadian DNA data bank uses the same system. So if you're looking for something a little bit more interesting this year for Christmas for somebody, you can get these DNA portraits. They're very nice. I encourage you to check them out. I don't know how much they cost, but they are very interesting, and of course every one of them is going to be unique. So let's talk about some medical applications of these DNA technologies. I mentioned this one here. I wanted to just go over it in a little bit more detail about how we can produce human insulin in E. coli. So if you take a look, there's a human cell and an E. coli cell. You can extract the DNA for insulin. So you can see we have extracted, here's the gene right here. And there's a plasmid DNA from E. coli. So just like the plasmid that we had in our lab, we can use the same one, in fact, to do this. They're combined together using DNA ligase, and this DNA is called recombinant DNA. Good definition to know for the final exam. We take the plasmid and we stick it back in E. coli, just exactly like we did in Lab 9. E. coli is put into a fermentation tank, and then it produces the protein. So there it goes, it's making human insulin. We can get that insulin from the media, and we can sell it. Here is the human insulin here. It's called humulin. Cute name. This has been done with many different types of proteins in biotechnology. Believe it or not, I was reading about a recent study where they had taken the mammoth genome and had started producing some mammoth proteins. The woolly mammoth, an extinct species, so very interesting. So here's something else that has been done. This is a company in the United States. It's called Genzyme. And so what they do is they specialize in making what they call farm animals. So I'll spell that for you. This is kind of a cute little thing. Farm. So pharmacy, or farm animals. Ha ha ha. Cute little word. I'll try to put this on the final exam somewhere so that you know what this means and you can get a mark for that. So what have they done? They basically try to put things in goats. And what are they trying to put in goats? They're trying to put this in female goats and have proteins expressed in the milk. So rather than producing an entire animal and then slaughtering it, you can actually have a protein produced in the milk. Why would you do this and not make it in E. coli? Well, it turns out that there are a lot of things that mammals do to proteins. We've got glycosylation and other things that are going on that E. coli doesn't do, and so they need to be made and produced in animals in order to get these useful products. So you make it in the milk, so you don't have to slaughter the animal. Here's an example. They've inserted the gene for human antithrombin 3. So what is that? That's an anti-blood clotting factor, and this is used in surgeries. It's a very expensive protein, and this company has found a way to produce it in goat's milk and can be sold to hospitals. Before this, how did we get this protein? We had to get it from human blood. So very useful idea. We also talked about these things before, these glowfish. You can go check them out at this particular website. These are fish that have the gene from jellyfish for fluorescence, and this is more of a novelty, but also something kind of interesting. Notice on the right here, somebody has made a glow-in-the-dark rabbit. Pretty weird stuff. I must emphasize that genetic engineering is not just for novelties. There are many useful things. So this particular project here, this is the Brainbow Mice project. So what were they trying to do? So it turns out that if you want to study the brain, traditional methods require you to dissect animals. And of course, this goes through a lot of animals. People are not happy when you're killing lots of animals in your research. And so what these particular scientists have figured out how to do is to add genes that express colors in these neurons. And so as the neurons are used, they actually light up fluorescently. And so what they can do is actually study brain functions in living mice, which is very fascinating. Saves a lot of mice and has really advanced neurological research quite a bit. So what about ancient DNA? Maybe you saw this little movie here, Jurassic Park, where you read the book, and you remember the whole principle here. This idea that, hey, we could get DNA out of amber, out of insects that lived in the dinosaur era, and maybe make dinosaurs. Turns out probably not. It doesn't seem the DNA lasts that long, or that we know of anyway, so we're probably not going to have a Jurassic Park yet. But there are some interesting cases where we have studied some old DNA, so I want to take a look at that. Here's an example where some ancient DNA was extracted from an individual. You may be familiar with this guy. You can see his name here. It's Otzi the Iceman. So Otzi the Iceman was found in the Alps. I believe it was between Switzerland and Italy, and he is the oldest, best preserved human specimen that we have. We do have older Egyptian mummies, but they're not preserved in such the same way. Egyptians were putting their bodies in chemicals and taking out organs. This guy was living during the Bronze Age, and we've learned all sorts of things. They've taken a look at his gut contents, and they can tell us what he's eaten. The interesting thing is here, they took a look at some of his DNA, and he was also carrying a bunch of arrows. And it turns out that the arrows had human blood on them, and they could determine that the human blood was not his own, and so that he had clearly lived somewhat of a violent life. There's a lot that could be said about this. I'm just giving you this as an example of how we are using DNA technology to do some archaeology. I'll show you another example. Here's another example of some interesting archaeology. This was about maybe 5 years ago. They found King Richard. I know you're probably wondering who King Richard is. If you're familiar with Shakespeare, then you know that King Richard was a hunchback, and he also died in battle. And in fact, historically, he was the last king of England to die in battle. They found his body underneath a parking lot. They excavated it. They were pretty sure it was his body. There was clear evidence that this person had a pretty protein-rich diet. There were some skeletal deformities showing he was hunchback. And they took a DNA sample, and it turns out there was a woman in Canada who was his distant descendant. And they tested it with her, and it was a match. And so they were about 95% sure it was King Richard, and after they did the DNA analysis, they were pretty sure that this was the case. Here's the evidence: the carbon dating, battle wounds, skeletal deformities. He was a hunchback, and like I said, the DNA sample was the last kind of checkbox to confirm that this was indeed King Richard. So what about those mammoths? I see this pop up all the time. Here's an old article I found. I grabbed it because it actually has some nice graphics with it. And you see this happening about every 2 years. I'll hear about people talking about the woolly mammoth, and maybe we could, use some Jurassic Park technology to make a living mammoth. There's been at least a couple Discovery Channel specials about this, and I'm sure something will pop up on Netflix someday. So one scientist said if he had $10 million, this is in 2008, he could make a mammoth. So how can we do this? Well, first of all, it turns out we actually have the mammoth genome sequence. This was done back in 2008. In fact, I think this has been done a couple of other times with other samples as well. From hair and various other specimens, we can take that and get the DNA sequence. It turns out, you can see here, it says it differs from the elephant genome by about 400,000 sites. So how can we make our mammoth? Well, we could use CRISPR or some other technology, and we could change those sites. So take a look. Here's the mammoth DNA up here. The African elephant, that's closer to the mammoth than the Asian elephant. And we have these different codons. So we have a CGT and an AGT. So we can just use a little bit of CRISPR technology, and we can change that. And maybe we can make a mammoth. Probably not as simple as it looks. We could certainly change the sequences in a genome 400,000 times. It would take quite a bit of finances, and of course, growing up in a surrogate mother, maybe an elephant, might be a lot more complicated than it looks. But in theory, we maybe have the technology to do this kind of thing, which is just really fascinating. I thought I'd show this to you guys. This is super cute. This was found only a couple of years ago. They found this baby mammoth, almost an entirely complete specimen. This was found in the permafrost in Siberia. Notice it's missing its tail, and that was pretty much it. And so this had blood in it and all sorts of other things that can be used to get those DNA samples. Here's something a little more scary. We could use DNA technology to make viruses and all sorts of things. This was done, take a look at the date. 2002, scientists created a live polio virus just using sequences off the internet. We can't do this with all viruses. Smallpox is a lot harder, and Ebola is a lot harder, thankfully. But this does lead to some implications of these technologies. Kind of scary stuff. So what about genetically modified foods? This here, by the way, is Photoshop. Don't believe everything you see. But I just want to talk about genetically modified foods for a minute. So GMO, often when people will talk about GMOs, and usually they're talking about genetically modified organisms or genetically modified foods. Here was the first genetically modified food. This is the Flavor Saver tomato. So it turns out tomatoes actually have genes in them that tell them when to ripen and spoil. So what they did was they tweaked these genes, they deleted some of them, and the tomatoes could be picked off the vine red instead of green. It turns out this was a bit of a failure because they were very expensive and people don't want expensive tomatoes. But take a look at the date, back as far as 1994. I'm sure most of you were not even born. So what about something a bit more useful? This is golden rice. This is a really fascinating example of a really interesting and good use of genetically modifying technology. So it turns out that many people on this planet live off of rice as basically a staple. And rice does not actually have a lot of vitamins and nutrients in it. It's mostly just starch. So this guy here, his thought was that vitamin A, or at least a precursor to vitamin A, which is beta-carotene, grows in other parts of the plant. So why not express it so that it grows in the seed? And so that's what he had done. He played around with some promoters and some transcription factors, and he was able to express this beta-carotene in the seed of the rice. Just makes it kind of a yellowish color because it has a pigment in it. And the thought is that this could be grown all around the world and would prevent blindness in some children and a number of other vitamin deficiencies. The big problem with this is not every part of the world is receptive to genetically modified organisms. Again, I'm not going to get into this in a lot of detail, but there are huge differences in opinions on whether we should be growing these kind of foods. And so he's encountered quite a bit of resistance in different parts of the world trying to market his rice. And he's not even looking for profit, he's just looking to help people. I encourage you to drop by my office anytime next year, and we can talk about these anytime. Like I said, I unfortunately can't really get into these issues in a lot of detail. So I just wanted to point out, with this controversy of genetically modified organisms, we've been doing this for thousands of years, humans. Take a look at this particular plant here. This is Grassia oligerica. I guess that's how you say it. This is the wild plant. It doesn't look like anything special, right? But it turns out humans have modified this by artificial selection, just normal breeding. And what have we done? Well, we've made broccoli, we've made cauliflower, we've made Brussels sprouts, we've made greens, we've made kohlrabi, kale, cabbage. Yeah, all of these are the same species. We've been genetically modifying things for years. Are these things natural? Are these things even able to grow in the wild naturally? The answer is no. They require human intervention. Many of our foods are not natural anymore. They don't grow like the wild versions anymore. So one part of this argument is people say that genetic engineering is just speeding things up. Rather than taking thousands of years, we can do these things in a much shorter time. Anyway, just thought I'd leave that with you as part of the debate. So I wanted to just mention a few exciting CRISPR projects. This technology has gained so much press attention because of the amazing things that it might be able to do someday. Here's some examples of some projects that are actively being worked on right now. So notice this first project, producing hornless cattle. I know you're thinking, yeah, what's the big deal? Talk to some farmers. Cattle have horns. Horns are dangerous. And many cattle will undergo a dehorning process, which is also dangerous for the cows. They can bleed to death. It's dangerous for the farmer to do the process. So why not edit these the genomes of cows so that they don't produce horns. And so this was actually done. This was a couple of years ago now, and who knows, they may already be on your dinner plate. So what about engineering mosquitoes? Check out this Wired article here. This is talking about a project where people are trying to figure out how to edit mosquitoes so that they can't carry the organism that carries malaria. We're not sure exactly how many, but probably 500 million people are infected by malaria a year, and it's killing probably 1 million children a year. If we can get rid of malaria, I am all for all this genetic engineering stuff. What about treating HIV? HIV is a disease of the immune system. We could, in theory, make people's white blood cells immune to HIV infection. There's another article here. You can read all about it in Nature and see what people are proposing. But this may be a therapy. This may be a cure. Right now, there is no cure for HIV, but maybe we will have one soon. Who knows? Okay, so here's a sample gel. Just a reminder that you will see one of these on the final exam. It says, do all the children belong to the father? Let's take a look at child 1. One band matches. Child 2, we've got a matching band. And child 3, yes. Yes, yes, and yes. So notice over here I have another question for you. Why does child 1 only have one band? Good question. Well, it turns out sometimes people have 2 alleles that are the same. So this person actually has 2 bands, but they're both traveling the same distance on the gel. Just kind of an interesting question. So here's the end of the topic 19. I've abbreviated quite significantly. Usually this is about 2 lectures worth. I've kind of cut it down to about 35 minutes in total. Make sure you know some of these techniques. Okay, we talked about restriction nucleases. We talked about electrophoresis. We talked about PCR. Don't forget PCR. Huge, huge important technique. You will see it in other biology courses for sure. There's PCR right there. Don't forget. We also talked a little bit about some of the applications of DNA technology. The textbook has all sorts of examples as well. I encourage you to check them out. We are not talking about cloning this semester. Some semesters I talk about cloning. Interesting technology. We are not covering it at all for this semester. There's some terminology for you. All of these things you should learn what they are, because they are fair game for the final exam. Okay, I hope you enjoyed topic 19. Usually it's a lot more interesting in class because we spent a couple of lectures looking at it and discussing some of the applications and implications. But I'm sure if you take more biology classes, you'll hear much more about it in the future. Here are the study questions. Take a look at these. Some of them are a little bit harder than others. Like I said, we're not covering this in a huge amount of detail. Make sure you know these definitions and some of the uses of these technologies. I hope you have a great day. I will see you later.